First, we will focus on the physiologic anatomy of smooth muscle. Main location for smooth muscle is in viscera, blood vessels, and gastrointestinal tract. There are two types of smooth muscle: multi-unit and single unit. Multi-unit smooth muscle is made up of discrete, means individually distinct, separate smooth muscle fibers. Each muscle fiber is covered by glycoprotein layer, and each fiber operates independently of the others and innervated by a single nerve ending. Examples of multi-unit smooth muscle, they include ciliary and iris muscle of the eye, and also piloerector muscles. Unitary type of smooth muscle is also called syncytial smooth muscle or visceral smooth muscle. Unitary smooth muscle is actually a sheet or bundle of interconnected muscle fibers having a mass of hundreds to thousands of smooth muscle fibers that contract together as a single unit. The cell membranes in unitary smooth muscle, they are joined by many gap junctions through which ions can flow freely from one muscle cell to the next. Well, unitary smooth muscle is found in the walls of most viscera of the body, like the wall of gastrointestinal tract, bile ducts, ureters, uterus, and blood vessels. Here is a table of the smooth muscle characteristics where the two types they can be compared and contrasted. You see there are certain features which are common in the multi-unit and single unit, while some are differentiating features, like the number of the gap junctions in the single unit smooth muscle is much more than present in the multi-unit. And here you can see that the action potentials they can be produced spontaneously in the single unit, but that is not the case in multi-unit smooth muscle. And then there are some more differentiating features, such as you see the contraction in the unitary smooth muscle, this can be produced by stretch only, but not in case of multi-unit smooth muscle. Moving on to the structure of smooth muscle. Smooth muscle contains both actin and myosin filaments, but does not have the same orderly arrangement in the form of successive sarcomeres. So there are no striations seen, and for the same reason, it is called smooth muscle because it does not show any striations. In addition to actin and myosin filaments, intermediate filament, they can also be present in the smooth muscle, and this intermediate filament, when present, this is made up of a protein which is called desmin. And keep in mind that smooth muscle, it does not contain the normal troponin complex. Instead, it has another protein which is called calmodulin. Now, if we look at the arrangement of actin and myosin filaments, they are scattered among the actin filaments in smooth muscle. There are 5 to 10 times as many actin filaments as the myosin filaments. The actin filaments, they are attached to the dense bodies, which are also called plaques. Some of these bodies are attached to the cell membrane and others are dispersed inside the cell. And these dense bodies, they contain a protein alpha actinin, which is very similar to the Z line. Some of the membrane dense bodies of adjacent cells, they are bonded together by intercellular protein bridges. It is mainly through these bonds that the force of contraction is transmitted from one cell to the next. Well, most of the myosin filaments, they have a side polar cross bridges arrangement. It means that the bridges on one side, they hinge in one direction, and those on the other side, they hinge in the opposite direction. And then there are cavuli, which are small invaginations of cell membrane of the smooth muscle, and they are analogous to the T-tubules of skeletal muscle fiber. So, what is this calmodulin? You see, calmodulin is abbreviated for calcium modulated protein, and it is a protein which is present in all the cells and it binds calcium. This binding of calcium is required for the activation of calmodulin. To understand the mechanism of contraction of smooth muscle, you must know that contractile filaments are arranged to form a diagonal web under the cell membrane. And as a result of this arrangement, contraction takes place in all the planes. Physical basis of the process of contraction of smooth muscle is quite similar to that of the skeletal muscle because the same principles of contraction they apply to smooth muscle as the skeletal one. Also, the same attractive forces between the actin and myosin filaments are operative. Although the internal physical arrangement of actin and myosin, this is a bit different. Here on the screen, you can see the stepwise events during smooth muscle contraction. First of all, action potential causes the opening of voltage gated calcium channels. So, calcium ion enters cytosol of smooth muscle fiber. This calcium ion it binds with the calmodulin, making a calmodulin calcium complex. The calcium calmodulin complex then activates an enzyme which is called myosin light chain kinase. Now, this activated enzyme provides energy to the myosin head, as a result of which the myosin head is phosphorylated and it becomes active, leading to the cross linkage between activated myosin and actin and ultimately contraction. Source of calcium ions for smooth muscle contraction are extracellular fluid mainly and the sarcoplasmic reticulum, but this role is only rudimentary. The role of calcium ions it's very vital in the regulation of contraction as you can see in this flow chart. Now, the cessation of contraction is achieved by two ways. First is by the removal of calcium ions out of the smooth muscle fiber and back into the extracellular fluid or back into the cytoplasmic reticulum by calcium pumps. And secondly, an enzyme, myosin phosphatase, is required for the reversal of myosin head's phosphorylation. So, what does it do? This enzyme splits the phosphate from the regulatory light chain of myosin. And absence of this enzyme leads to the stoppage of attachment detachment cycling of myosin and actin. This is the graphical presentation of the process of contraction, and this part is showing the process of relaxation in the smooth muscle. Well, this is your university question. The latch phenomenon describes that when smooth muscle has developed full contraction, it continues to be contracted despite the degree of excitation has been greatly reduced. A smooth muscle can maintain prolonged contraction. Once excited, it remains contracted without further excitation. One example of the muscles in which latch mechanism is observed is the sphincters of gastrointestinal tract. Significance of latch phenomenon is multifold. The contraction is maintained for hours with little use of energy, and the rate of splitting of ATP by the cross bridges that also declines. And this occurs when a phosphorylated cross bridge it becomes dephosphorylated while still attached to actin. So the dephosphorylated cross bridge dissociates from actin by binding with ATP at much slower rate than the phosphorylated actin. Stress relaxation is another unique phenomenon which is seen in smooth muscle. So, when the smooth muscle is stretched, um, initially there is an increase in tension, much like the rubber band. But slowly, the tension it comes back to the resting level due to the readjustment of cross bridge attachment. So, the phenomenon of stress relaxation is achieved by the readjustment of the cross bridge attachment. So we can say that the smooth muscle it exhibits a phenomenon which is called stress relaxation response, in which the smooth muscle it responds to stretch only briefly and then it adapts to its new length. The new length, however, it retains its ability to contract. And because of this phenomenon, it enables the organs such as stomach and bladder to temporarily store the contents. Keep in mind that the skeletal and the cardiac muscle they respond to stretch with a more forceful contraction. Main aspects of comparison between smooth and skeletal muscle contraction are listed here as you can see that our smooth muscles they show slow cycling of myosin cross bridges and low energy requirement. The slowness of the onset of the contraction and relaxation in the smooth muscle that is another differentiating point. And our smooth muscle it also exhibits stress relaxation when it is needed, and also the latch mechanism or the latch phenomenon. And these phenomena they are not seen in other kinds of muscles. Let me now tell you the electrical properties of smooth muscle. Resting membrane potential of smooth muscle generally lies between minus 50 millivolts to minus 60 millivolts. And you already know that the smooth muscle, this is of two types. So we will discuss the uh, depolarization in smooth muscle according to its type. First, let's understand the depolarization in multi-unit smooth muscle. There is only localized depolarization in multi-unit smooth muscle which results into contraction. It is postulated that no typical action potential is produced because the muscle fibers are too small to produce action potential. In other words, in multi-unit smooth muscle, local depolarization takes place without action potentials. And instead, the chemical transmitter substances cause depolarization, which in turn elicits contraction in multi-unit smooth muscle. How does unitary smooth muscle depolarize? In unitary or visceral smooth muscle, 30 to 40 muscle fibers must depolarize simultaneously to produce an action potential. For the smooth muscle contraction in the unitary muscle, the same sequence of basic events takes place. And then you should also know that which nerves supply smooth muscles. You see, smooth muscle is innervated by autonomic nerve fibers that branch diffusely and excrete neurotransmitter from multiple varicosities, which are actually the localized nerve fiber swellings serving for motor end plates. The vesicles of autonomic nerve endings, they contain acetylcholine in some fibers and norepinephrine in others. And occasionally, other transmitter substances, they are also present in the autonomic vesicles. Neuromuscular junction of unitary smooth muscle is called diffuse junction in this unitary smooth muscle, and it is not of a highly structured type which is found in the skeletal muscle. The transmitter substance which is secreted into the interstitial space that diffuses directly into the muscle fiber. In multi-unit smooth muscle, neuromuscular junction is called contact junction. Here, the varicosities are separated from muscle cell membrane by only a 20 to 30 nanometer width of synaptic cleft. Major neurotransmitters released at neuromuscular junctions in smooth muscle contraction are acetylcholine and norepinephrine. Both acetylcholine and norepinephrine excite or inhibit smooth muscle. When acetylcholine excites, norepinephrine ordinarily inhibits the muscle fiber. Conversely, when acetylcholine inhibits a fiber, norepinephrine usually excites it. So, the type of the receptor determines which of the two transmitters is effective in causing the excitation or inhibition. As I already told you, that the resting membrane potential of smooth muscle is minus 50 millivolts to minus 60 millivolts. So you can note that the resting membrane potential in smooth muscle is about 30 millivolts less negative than in skeletal muscle. This resting membrane potential leads to slow wave potential. Now the question is that what is slow wave potential? In visceral smooth muscle of intestines, resting membrane potential becomes more or less negative alternatively. And this is called slow wave potentials, slow wave rhythm, or the pacemaker waves. Slow waves in resting membrane potential. It is a local property of unitary smooth muscle fibers. Normally, membrane potential keeps fluctuating between resting membrane potential and the slow wave potential all the time. So, how the slow wave potential is different from the action potential? You see, the slow wave itself, it is not an action potential, but it can give rise to action potential. And then the slow wave potentials, they fire the action potentials only when they reach the threshold. Slow waves themselves, they cannot cause muscle contractions unless they generate action potential. The cause of rhythmicity in smooth muscle is the waxing and waning of pumping of sodium ions outward through the membrane of fiber. And it is a self regenerative process which spreads progressively over the whole membrane. So, when you are asked in your Viva question that what is the significance of slow waves in the resting membrane potential of smooth muscle, your answer will be that this kind of potential it leads to spontaneous generation of action potentials in the unitary muscle, and hence it makes the unitary muscle self excitatory. In other words, you can say that it causes pacemaker activity and hence the rhythmical contractions in the unitary smooth muscle. Example of slow wave potential could be peristaltic activity in gut. In unitary smooth muscle, depolarization leads to action potential, and there are two types of action potential in visceral smooth muscle, a spike action potential and an action potential with a plateau. In most smooth muscles, stretch, that is the main stimulus. Spike action potential is the typical smooth muscle action potential. Single spike can be elicited by an external stimulus or repetitive spikes by slow waves, as in the intestinal wall. And the duration of spike action potential, it usually is from 10 to 50 milliseconds. Stimuli for spike potential could be stretch, hormones, action of neurotransmitters, and even the electrical stimulation, as in case of experiments. Spike potential is produced when the resting membrane potential it rises to minus 35 millivolts. It means when the slow wave potential reaches minus 35 millivolts, there is a spike potential that results in muscle contraction. The channels involved in depolarization are slow sodium potassium channels. At the termination of spike potential, slow wave potential again becomes more negative automatically, means it goes to minus 50 millivolts. Peristaltic wave in the gut, it is set up because of the stretch by filling of the gut with intestinal contents. The mechanism it involves the combination of two factors, the normal slow wave potentials and a decrease in overall negativity of membrane potential caused by stretch. Now, the plateau action potential. The onset of this action potential with plateau that is similar to that of the typical spike potential. The importance of plateau is that it can cause prolonged contraction, which occurs in ureter, uterus, and even in vascular smooth muscle. Phases of plateau action potential, they are rapid depolarization, then a plateau, and finally a repolarization. And the plateau, it can last for several hundred to 1,000 milliseconds, mean for whole 1 second. Control of smooth muscle contraction by non-nervous factors is achieved by local tissue factors and hormones which may cause smooth muscle contraction with or without action potentials or without nerve stimulation. As a matter of fact, without action potential, contraction is brought about by the entry of calcium ions into the cell upon depolarization. Uh, Here are the examples of local tissue chemical factors, which include lack of oxygen, which results in smooth muscle relaxation, and other examples they are excess of carbon dioxide and increased hydrogen ion concentration, both of which they result in relaxation. But how this relaxation is achieved? Actually, these factors they cause the hyperpolarization inside the muscle cells. And other local factors which affect smooth muscles, they are adenosine, lactic acid, increased potassium ions or diminished calcium ions, even increased body temperature and decreased blood pressure can also affect smooth muscles. Hormones also have an effect on the contraction or the inhibition of contraction. You see, the contraction is achieved through hormone gated excitatory receptors because these receptors they cause the release of calcium ions from internal sources without any change in the membrane potential. Then the inhibition of contraction that is brought about by hormone binding with the inhibitory receptors, and these inhibitory receptors they activate certain enzymes in the cell membrane. Here is a list of the examples of hormones and neurotransmitters that affect smooth muscle activity. But keep in mind that smooth muscle, it shows diversity in response to chemical factors. What it means that the same substance, it may cause either relaxation or contraction of smooth muscle in different locations by interacting with diverse receptors. For example, norepinephrine, it inhibits contraction in the intestine, but it stimulates contraction in blood vessels. Now, students, here is a list of the differences between skeletal and smooth muscle. And my dear, you are supposed to memorize these for your exam question.